ever wonder how food manufacturers come up with the nutritional information on the side of food packets? For example, how do they know just how much fat or sugar is in a chocolate bar? The answer is that they employ food scientists who carry out tests on the foods. You will have carried out similar tests using chemicals and equipment in the science lab. In your lab, you wouldn't have been able to measure the exact amount of nutrients in food. These are called quantitative tests, the kinds performed by professional food scientists. However, you will be able to tell if a food contains certain nutrients or not. These are qualitative tests.
When carrying out food tests, it is important to have a results table ready to put your results in. This table will list the foods down one side and the nutrients you are testing for along the top. A tick can be added to record which nutrient each food contains.
Just like any experiment you carry out in the lab, it's important to carry out a risk assessment before starting. Some of the chemicals used in food tests and the equipment you use have the potential to cause harm. These are hazards. Before you start using chemicals or equipment, you need to be aware of the hazards, the possible risks, and how to reduce them. The risk is the potential that someone could be hurt from the hazards.
There are three main food molecules that these tests are used to detect: carbohydrates, including starch and sugars, proteins, and lipids. Starch is a type of carbohydrate found in plants. To test for the presence of starch, you use iodine solution, which is coloured orange. The food to be tested is placed in a dimple of a spotting tile, and a few drops of iodine are added. If the food goes blue or black, starch is present. The presence of a blue-black colour is a positive result for starch.
The iodine you use will be a very dilute solution, so it is a low hazard. However, you should still wear eye protection when using it, clear up any spills immediately, and be careful not to get any on your skin. It's worth noting that chemists use starch to test for the presence of iodine by reversing this test.
Some sugars are reducing sugars. Examples include glucose and fructose, the sugar found in fruit. Reducing sugars are tested for by using Benedict's solution. This blue solution will go orangey-red when heated with a food that contains reducing sugars.
It is advised that you use hot water from an electric kettle for the hot water bath. Do not use a Bunsen burner to heat it. This is because of the risk of ethanol, perhaps being used elsewhere in the lab in a lipid test, being exposed to the flame and catching fire. Using hot water carries risk of burns, and also you must be careful when using electrical equipment around water. Mixing water and electrical equipment can cause electric shocks.
Benedict's solution is a low hazard. However, you must wear eye protection when heating it because of the risk of spitting when it is heated.
To test food for protein, you would use pale blue Biuret solution. If the food is solid, you will get a clearer result if you grind the food up with some water in a pestle and mortar and decant about 1 cubic centimetre of the liquid, which is about a fingertip's depth, into a test tube. Add about the same volume of Biuret solution, give it a shake and leave for a few minutes. A positive test for protein is a purple colour.
Biuret solution is a mixture of copper sulfate solution and dilute sodium hydroxide, an alkali. It is therefore classed as an irritant. This will be indicated by the hazard symbol of an exclamation mark on the bottle. You need to wear eye protection when using Biuret solution, and you must wash splashes off your skin immediately.
Lipids can be tested by using ethanol. If you grind the food up with ethanol and then add the mixture to water, a cloudy appearance tells you that lipids are present.
Ethanol is highly flammable. This is shown by the hazard symbol of a flame on the bottle. There is a risk of it catching fire, so no naked flames should be used around it.
It's not just foods that can be tested by using these methods. The presence of glucose and protein in urine is an indication of illness, so it's important that doctors are able to test urine for these molecules. However, as these tests take time and require a lab to carry them out in, sticks have been developed that contain reagents. These can be dipped into a sample of urine. A change in colour will quickly indicate the presence of protein or glucose. This method is much preferred by doctors, as in the past, the only way doctors could test for glucose in a patient's urine was to taste it to see if it was sweet.